Shortly about myself, I'm vice chair of ERGA. I think you heard like a bit of who I am and what am I doing, but I have many different hats on me. One of them, I am coordinating biodiversity genomics project at the Swedish National Forest, the SciLife Lab. So I'm going to talk to you a little bit about the sequencing facility perspective. So what is happening? Especially, I was really triggered by a comment on the one of the meetings. So why can't you sequence that? It's A, G's, T's, and C's. So I will try to answer that particular question today, and also will give you some information. Why is it so important to keep a contact with the sequencing facilities? So the sequencing part of the reference genome project. Because of the limit of time, I will not go into all the possible technology and sequencing recipes and how can you do different things. Recommendations about the sequencing recipes you can find on ERGA and BGE homepages. But very shortly, for sequencing of the reference genomes, we are dependent on two major technologies. It's either Pacific Biosciences with high rates, high fidelity rates, achievable on the radio instrument, or we can use Oxford Nanopore with Promethean on Redine instrument. So just to give you a very short, brief introduction into how these instruments work. So hopefully it can clarify some of the issues that you might have with non-model samples. So the radio instrument, it came just a couple of years ago. It runs for 24 hours. And company will tell you, if you look at their website, it can produce up to 100 gig of data, 7 million reads, and so on and so forth, which is bull. That is achievable only on human samples, very seldom in vertebrate samples. As soon as you move one step aside from a vertebrate, from a plant, if you go into more and more weirdness that ERGA and BG are sequencing at the moment, the less data you can count in the getting. So getting 98 gigabases of data per sequencing cell, that is a very rare case of BG and EPP. So first take home message, never trust the vendor. How does PacBio work? So when the sample is coming to the sequencing center, first of all, we need to assess its quality. So the length of the fragments.
Sorry, Olga, can I interrupt just 1 second?
Yes.
Because for me, your audio is kind of fuzzy, and I see other people are also struggling a bit with the quality. I don't know if there's something. Can you hear me now? Yes, yes.
Should we do it without audio?
That's much better for me, yeah.
Okay, if everybody is okay with that, let's go on. So,
thank you so much. Sorry for the interruption.
Yeah, no worries. So PacBio is something that we are calling smart sequencing. It's not just because it's a very smart way to do that, but it's single molecule real time. When the sample comes to the sequencing facility, first of all, we need to assess the fragment length. So the good intact DNA usually has a peak of fragments over 40 kb. And the more of such fragments we have, the more happy we are and confident that this sample might actually work. After that, we do a fragmentation, and we want to fragment our samples or DNA molecules to a specific type or length. Usually it's between 15 kb and 20 kb. So this is our double-stranded DNA molecule. And you can see on the slide here that in PacBio, we are ligating 2 hairpins. Virtually, we want to circularize this molecule. So then we ligate a polymerase. Polymerase will sink to the bottom of the ZMV or nanowell present in the smart cell of the sequencing. And then polymerase will stick there at the bottom and hold into the DNA molecule. You start giving polymerase some food and some cofactors and nucleotides. Every nucleotide is labeled by different colors, and then the polymerization will happen. So polymerase sits there at the bottom and starts dragging DNA through itself. Polymerase is modified 529. It's very sloppy, but it's fast. When it goes from one hairpin through the molecule to the reverse hairpin, the error rate is pretty high. It can be around 10%. But then it goes through the hairpin and reads the molecule into the reverse direction. And because the error is random, it corrects itself. So when you have one strand and the second strand, you can see here for a slide. So there are some errors here, other errors there, and then polymerase will continue in the circle, reading forward adapter, forward reverse, forward adapter, reverse adapter, forward adapter, reverse adapter, and so on. So, and then bioinformatically, you'll stack all these reads on one another. You'll make the error correction, and at the end, you'll get a beautiful hi-fi read. Hi-fi read is quality of Illumina-like, but polymerase translocates with a speed of 3 nucleotides per second. That means that 15 KB library in 24 hours that we're going to run the machine, it will be read many a time. So finally, we're getting to the quality scores of sometimes 70, sometimes 80, or even 90. So we can proofread both Illumina and Sanger sequencing using the PacBio data. This is how the hi-fi reads are generated. All right. Next, we want to look at the Oxford Nanopore, which is another technology. Oxford Nanopore does a completely different type of sequencing. It's not based on synthesis. So in Oxford Nanopore, we have different instruments from the smallest, Flongle, that we usually use for QC of the libraries. You can also use it for bacteria. Then we have MinION and GridION. GridION is pretty much 5 MinIONs plugged in the same way together. So it's 5 MinIONs working in parallel. And then we have the PromethION, where flow cell is a little bit different. It has much more pores, so it can produce much more data. And as you can see here, I have lots of asterisks all over the chart. So we have output per cell, maximum number of reads, and also read length. Very often when you read what company is telling you, yes, we can produce 4 or 5 megabase long reads. Right, but you cannot produce your 5 megabase long reads if you do not have megabase size molecules in your prep, right? So it depends on the quality of DNA that you are getting. In Nanopore, what they say about the PromethION, that you are able to get up to 290 gigabases of data, I have never seen it in my life. Maybe in human cell cultures, yes, I have seen that other centers are reporting that. We are standardly or usually getting around 120 gig on human data. But as soon as we're trying to do on something else that is non-human, we can get, well, in a good case, perhaps 40, perhaps 60 gigs. So again, forget everything that companies are telling you. Also, Nanopore, in some biodiversity genomics projects, it will clog the pore. The pores will die. The reaction will be slow, and so on and so forth. So nothing works for biodiversity genomics projects as the companies usually advertise. How does the nanopore sequencing work as compared to the PacBio? So if you can see from the slide, we do not circularize the molecule. We introduce only one hairpin. So for the machine to understand that this molecule has to be sequenced, we need to ligate 2 adapters that are recognized by the pore. And then we need to put a motor protein. This is something that will be sitting on the pore and will start unwinding DNA. Then we're introducing cholesterol molecules that will weigh down the DNA molecules to the membrane where the pores are. And then you have pores. Imagine yourself a protein channel that is going through the biological membrane. So it's DNA is coming to the pore, then adapter is being recognized, so the DNA is dragged to the pore, and then on top of the pore, the helicase or the motor protein will be sitting. And then it will start unwinding DNA, and the DNA will be dragged through the pore. In the pore, while DNA is being dragged through the pore, right? So there is a voltage change because there is a potential change between 2 surfaces of the membrane. And when different bases are passing through the pore, they change the voltage. And that is something that is recorded by semiconductors, which then translate these different squiggles of voltage changes into the bases, right? Nanopore still is a little bit sloppier than PacBio, but I must say that the recent pores, they're much better, producing much better quality of data, because you have two different points now when you can read out the voltage changes. So it's a little bit of auto-correcting itself. And there are also duplex reads that many people heard about. The problem is that when you're trying to sequence in the duplex mode, you're getting much less data than you would normally expect. So people usually ask, so what do you think is the best technology to sequence your reference genome on? I would say go by proximity, which sequencing center is closer to you. Because for PacBio, we usually say we need 15x coverage per allele, whereas in Nanopore, we usually say 30x coverage per allele is needed, right? But here are these two technologies, and now you know how they work. But why is that question about the A, G's, T's, and C's, and why my DNA does not sequence? And here we are coming to the DNA quality requirements. There is a mantra around the sequencing facilities worldwide. So garbage in and garbage out. So the sequencing success always, I would say to 90%, depends on the sample quality. And another very important thing to say that next generation sequencing or long read sequencing quality DNA is absolutely not the same as a PCR quality DNA. And I will show you why. This is an example that I use a lot. This is one of the first runs we ever ran on PacBio. We got it in the early 2013, and that was one of the first runs that we ever performed. So this is the upper part of the slide. So if you can see here, these are the statistics of the data that we got from that particular sample. It was a yeast. The genome size was around 7.8. It would have 8 chromosomes and the mitochondria. When you look at the N50, I appreciate that in this auditorium, perhaps not many people know how to read the statistics data of the assemblies. To say it simple, so if you have different reads, if you can overlap them, that means that they're hanging together. So that means from here to here, you can concatenate these reads through the overlap, and that would be your contig. The more reads you can stack, the longer contigs are going to be. So N50 tells you that in your assembly, the majority of your contigs are of this size or larger. So N50 should be large. For BGE project, for ERGA, it should be in megabase scale. So here you can see we had only 2K. Also, it was very fragmented. We had lots of different contigs, and we didn't even approach the genome size. At the same time, a couple of months later, we got another strain of exactly the same species that came from a different lab. And what you can see here, we had 9 contigs. So we have 8 chromosomes and the mitochondria, all of them in one beautiful stretch of nucleotides. And 50 of the assembly is a chromosome scale. So 50% of the data, they are in this size contigs or even longer. The maximum length of contigs is again a chromosome, and the sum of contigs, that is your genome size, 7.8. And we're like, okay, what is going on? We are using exactly the same reagents, the same machine. How come these two different strains, they are so different when it applies to sequencing? And we digged wide and everywhere. And the only difference between these two, that was actually on DNA. We checked the absorption ratios here. It was very poor. And you can see there is some degraded DNA and something else in the sample, whereas DNA for the second good strain, it was extremely pure, very beautiful, and it worked like a charm. And that was actually a wake-up call. And both myself and colleagues from Sanger and Oslo, we started to follow these things up. And we started discovering things that companies were not sharing or were not really very widely known. So in the long-read sequencing, unlike in Illumina, we're sequencing native DNA molecules without any PCRs. These samples work in Illumina because Illumina is PCR-based. So still you can salvage some molecules and those will be sequenced. In PacBio and in Nanopore, we do not have this pre-treatment step with the PCR. So we're sequencing the sample as it is. And there are tons of different things that can react as inhibitors of the long-read sequencing. That can be compounds that are carry over from the cells where we're doing our instructions. Those can be proteins. Those in plants, they can be polyphenols. In fungi, different amphibians and different insects, they can be toxins, they can be secondary metabolites, they can be pigments, polysaccharides, and so on and so forth. Some of them, they will covalently bind to DNA molecule. And in PacBio and in Nanopore, we have isothermal amplification. So we are running that at ambient temperature or at 37 degrees. And 37 degrees is not enough to denature a molecule that has some sort of contaminant on it. On Illumina, it's 92 or 95, so it's fine, open. When polymerase is coming to some complex that cannot open at 37 degrees, the polymerase will not pass and you'll have a short truncated read. Or polymerase will sit there and stutter, and then maybe it will jump. But it's still quite an anecdotal evidence, and we definitely need to make much more R&D to understand this phenomena of how PacBio and Nanopore are single-strandedly sequence the contaminated molecules. So it's a very difficult issue. Besides different types of DNA binders, there are tons of different polymerase inhibitors. So for instance, EDTA, if you have too high a concentration of EDTA in your buffer, EDTA will scavenge all the cofactors that are needed for polymerase and for helicase to work. Phenol, it will denature the polymerase. That is, if you are doing phenol chloroform extraction and do incomplete phenol remover. And then if you have alcohol and so on and so forth. Or if you have some debris left in the DNA extractions, that will just physically interfere with both library preparation reaction and with the sequencing. So optimally, what we would like to do, we would like to start working with the protoplasts. So as we humans have like nice naked bodies and no nasty cell walls. And then from this protoplast, we will just extract nuclei and DNA. Well, it doesn't work that well, doesn't it? So we have different types of cell walls that are present out there. So look, for instance, in fungi. So these are the chitins and proteoglycans and other things. So they will be there to contaminate your DNA. Same in plants, that is completely different story, polyphenols and so on. And then in insects, you have the carapax or the outer shell, or you call it like this, and so on and so forth. So imagine you are DNA, you are sitting inside the nucleus, inside the cell, you have all these beautiful proteins that keep you nice and cozy, pH is fine, all the reactive oxygen species are being taken care of, peroxisomes and other enzymes and so on. And then you open everything up. So the content is leaking out. The reactive oxygen species attacking all the molecules that are there. So there is hydrolysis. Nucleases and everything else. Everybody's trying to attack the poor DNA. So, for instance, in case of plants, when you will not add your antioxidant like beta-mercaptoethanol to the DNA extractions reaction, what will happen, the polyphenols, they will oxidize. And when they oxidize, they bind to the DNA, cross-linking it and making that part of DNA completely unsequenceable. There is no way to remove those reagents after they got attached there. So things like that, all these small different things can make our life as a sequencing facility and you as a scientist very, very complicated. In 1983, there came PCR. So people pretty much forgotten how to do old school phenol chloroform extraction. And now people are trying to move away from these highly toxic phenol extractions. So we need kits that work. I can tell you from experience, everything that is called quick and easy and fast will not work for high molecular weight DNA extraction from non-model organisms. Here on the slide, I summarized some of the kits that work really well. Unfortunately, none of the kits work for everything. For fungi, nuclear spin from masherine gel might work really well. For some tissues, Monarch would be absolutely the best option. For some of them, like insects, genomic tip series or Magatract do usually a very good job. But you cannot just buy one kit and hope that it will solve all of your problems, especially if you are working in different taxonomic groups, right? But finally, somehow you manage to get DNA. So what is going on after? You need to make a quality control, at least something that can predict if your DNA will work or not. The one assay that is used the widest, I would say, this is a spectrophotometry when you're looking at the absorption ratios. So DNA, pure DNA, it absorbs at 260 nanometers of the wavelength. So we want to compare this absorption at 260 with other most commonly known types of contaminants. So 280, that's a majority of organics, like proteins and phenols and something else, that would be photoactive there, right? So if you have too low a number of 260 to 80, that means that you have too much of organic contamination as compared to your DNA. If you have it much higher, that means that you have a higher proportion of the RNA, for instance. 260 and 230 is way more tricky because there is salts, there are peptides, humic acids, some aromatics, and so on. So when it is below 2, that's pretty easy. Most likely that sample will not work without a good cleanup. But if it is higher than 2.2, I could not find any good comprehensive information if that would somehow affect the reaction or not. So that is a very good first check to understand if your DNA is pure enough. But what happens if it's not? Don't despair. There are different ways to fix it. So first of all, there is QIAGEN DNeasy Power Cleanup Pro, and that is called MoBio Cleanup Pro. It's a very good kit that can salvage a lot of dirty DNA. There is also another kit from Zymogen that is called Genomic DNA Cleanup and Purification Concentrator Kit. We also seen on some genomes it performs miracles. On others, it does not. Very often people are coming to me and say, Olga, I use DNA Power Easy. I lost 80% of my sample. I say, well, these 80% of molecules will not be sequenceable anyway. So at least you manage to get 20% of molecules that have a chance to be translated into the sequencing data. So even if you use lots of material, still you're saving your sample, you're saving your genome. In other ways, if you don't want to work with kits, you can also purify your DNA with different types of SPRIs. So for instance, impure beads, you can add extra phenylchloroform cleanup steps that we do a lot for plants and for the mollusks, for instance, for fungi, something with lots of sugars and so on. And we, both Sanger and us and other sequencing centers, we're trying to dump all those protocols into the protocols IO or anywhere at the open access. Another thing is when the user is not sending us a tissue, but sending us a DNA, they say, oh, I sent you 10 micrograms. And after our QC, we see it's perhaps only 1.5. And they get extremely frustrated. So why has DNA degraded during the transport? Well, it's not that easy. Again, during all these ages of PCRs and quick and easy things, we have forgotten a lot of classical biochemistry, right? So I've seen PhD students taking out DNA, frozen DNA from the fridge, throwing it on the table, and then just taking the sample out, and then they are measuring the concentrations of nanodrop. The problem is that when you're thawing DNA, DNA thaws at the gradient. So you have very low concentration here, a very high concentration here. And if you take it here from the tube, from one place, second place, third place, fourth place, you'll get 4 different concentrations. So, and which one is correct, right? So here, and these slides will be available to you after my talk. So here is a recipe what SciLife Lab Genomics is using for initial estimation of DNA amount. That is, how do you need to treat your DNA before you actually take the measurement. Another point I want to make, that nanodrop is bad. Why is that? I just said it was good. It was good for one thing. It's good for your absorption values. Why is it bad? Because nanodrop goes for everything that absorbs at 260. And that can include also degraded DNA, nucleotides, some RNA molecules, and so on. So what you're measuring is not a pure DNA per se. You're measuring all the soup that absorbs at 260. And unfortunately, it's not just DNA. What we do at sequencing facilities, we usually compare the spectroscopy or absorption readings with a qubit. Qubit uses a fluorophore that binds only to the hydrophobic part of the DNA molecule. So it measures only the concentration of double-stranded intact DNA. So that is why it's so good to have estimation of nanodrop and estimation of qubit. So you know how much total you have in there and what fraction of that is molecular weight DNA, right? So, and we always recommend to compare the nanodrop and qubit readings. And if you see that there is more than 50% discrepancy, it's a huge warning red flag that something might go wrong in sequencing reaction. The problem is it's not 100% safe. So we've seen samples where the skew was even higher and they worked fine. But for the majority of samples, that is actually stands true. If there is a more than 50% difference, more than double reading, that nanodrop says there is double as much, you might be in trouble. So such a DNA should be purified. If you want to achieve long libraries, long, beautiful nanopore reads, you really need to also think about DNA degradation. So when DNA is in the solution, you treat it as a crystal vase. You don't drop it on the floor. You do not vortex it. You do not pipette it too vigorously. You do not dissolve it in water. Please stop doing that, especially if you want to put it in the freezer. TE buffer, low TE buffer. You need something to buffer the pH so that to prevent DNA from hydrolysis, which will happen even at -20 degrees in the freezer. If you keep the DNA in water for 20 years in the freezer, it will start getting degraded. So be very careful. Use -70, use TE buffer. Minimize freeze-thaw cycles. We tested 5. That was not a big deal. So up to 5 freeze-thaw, it's still okay. And here in the corner, I made, together with the one PhD student who made a short study where we dumped in bioRxiv because none of us had energy to publish, but there is some nice review information for you if you want to follow it up. So finally, in the final couple of minutes, I will give you some real-life examples of quality versus sequencing. So these are the samples that we're usually dealing with and how we tackle the troubleshooting. So that was a pretty easy example. That was also very early. It was somewhere in 15. So it's a vertebrate, birds have nucleated blood, everything is fine. But that was here was the first QC that the PhD student sent to me. And I said, dude, this will not work because DNA is degraded, absorption ratio is out of whack. He never done this kind of DNA extractions before. So we were communicating in emails. So altogether, I calculated 74 emails. And this is here with a bottle and a saber. That is how you make a wide bore pipette tip, 5 o'clock in the evening on Friday, when your shipment of wide bore pipette tips did not arrive yet. So But finally, within a month, he managed to get this beautiful DNA, and we sequenced that particular crow, and it's beautiful. So the N50 of the assembly is over 10 megs. And also you can see the DNA quality was very good. We had some DNA fragments over 100 kb and so on. We could do pretty much anything we wanted with that DNA. That was a PhD student. Here is another example of a PI who is very well renowned, will not tell you the name, will not tell you the country, will not tell you the species name. The PI pretty much dismissed all my attempts to tell him that his DNA is not good, because that worked for Illumina. Well, the library was 8 kb, and we didn't get any context, we got scaffolds. And you can compare the N50 that he got with the N50 that person who listened got. So there is quite a big difference in quality of the genome, depending on the quality of DNA that one gets to work with. Example with beetles. Again, this is 2016. That is before we got our own DNA lab. So here I spent around half a year with a postdoc who was doing the extraction. So we tried different types of iterations. So DNA was either dirty or it was fragmented and so on. So we tried different types of iterations for protocols. Finally, it's beautiful reaction, beautiful sequencing. And there are 2 papers coming out of that, and I'm very happy about that. But again, half year to troubleshoot DNA extraction. Another sample, the same species, male and female, precious Australian samples, very large insects, quite a large genome. In case of insect 1, we could extract DNA ourselves. It was beautiful and everything worked fine. So this is insect 1. You can see the Contigan 50, everything is okay. In case of the second beetle, unfortunately, we had to work with Australian DNA. So, and the quality was not superb. The reads or the library we got was quite short. So, and then again, the quality would never be as good as from a nice, fresh, frozen sample. Very last example that I want to show you, these are the plants. And that was completely unexpected. So we were running some projects with a company in France, and they are really, really good in DNA extraction. So we always got a beautiful DNA sample from them. You can see this is a pulsed field gel. Absorption spot on. DNA is long and beautiful, high molecular weight. We were very happy. We're starting to work with that. We fragment it. It just falls apart to 2 kb. I said, what the hell? We're doing something wrong. So here, for instance, here is non-shared DNA and here is shared DNA. So it's a complete smear. So then we tried several different samples in parallel and applied exactly the same conditions. This is a good sample, this is a semi-good sample, and then that particular maize sample, and everything is just falling apart. So we just created a couple of crazy hypotheses. First, we wanted to check if the DNA was pure, and it was. So we thought either we have nicks on DNA, or we have abundance of transposons or low complexity regions, because those can also contribute to the stability of DNA, physical stability of DNA. So we introduced several steps of the DNA repair and so on. And finally, we managed to get 10 kb libraries, but not 20. But when bioinformaticians started to look at the data, there was an even transposon insertion pretty much every 5 kb of that particular strain. So possibly or probably that was a combination of both. But that's a problem when you are in the sequencing center. You solve your problem, you go to the next genome. Solve your problem, go to the next genome. You never get a possibility to look at the biology of species and put together in one sack and then get an overview of, did my hypothesis actually translate into the biology of species? But in that particular case, at least we have an indication that it could be due to the transposons. Besides the different DNA types of DNA quality samples, they react in different ways when you try to do sequence or extractions. The machines, both PacBio and Nanopore, they work in their mysterious ways. So here's another example. This is a salamander. I can tell you that. It's a large genome. And this is exactly the same DNA. We just did 4 different libraries on that DNA. We have standard operational procedures that were prepped in parallel. We have extremely tight quality controls all the way until the sample goes to the machine. But if you look at the yield per smart cell on the same DNA, you can see that the difference sometimes is double, like 32, 33 here and 15 or even 12 here. And then the length and number of passes through the same molecule per cell would vary a lot. And also the length of the reads, the hi-fi reads that we're getting. So I wish to say that PacBio is a very nice and robust technology. We like it, but sometimes it's not as robust as we would like it to be. And same goes for Nanopore. And the other slide, it's courtesy of James Watt from the Sanger Institute. All these pillars, what you see, they're different taxonomical groups. At 60 kb, there is a cutoff of what we consider a good loading or good result, sequencing result from a smart cell. And then you can see that these are different species. And it's all over the place. This is the same taxonomic group, and some of them sequence really well, and some of them do not sequence well at all. Even more so, sometimes when we have too little of material or DNA is so contaminated we cannot deal with that, we introduce a PCR step. It's so-called ULI, or ultra low input library. Even on ultra low input libraries, you see that for some taxa, still we get underloading and still we get much less data than for other types of libraries. And it's virtually impossible to predict. This is something that we have communicated both with Nanopore and with PacBio. We want to have a single analytical parameter that will tell us that it has a predictive value to say if the sample will work or the sample will not work and how it will work. Up to now, with Sanger, with Dresden, with Oslo, almost 10 years now, we are trying from thousands of samples we have sequenced to find any sort of correlation. We cannot, and companies cannot. So the only thing they could help us with was the adaptive loading, but that's a thing for a different conversation. And just before I let you go, so the practicalities. So sequencing facilities, when you start your reference genome sequencing project, get in touch with us before you go to the field, because based on our experience, we can advise you how much material do you want to have. However, that also varies between different centers. So I would suggest choose your sequencing provider. There will be information about that in ERGA, or ask around and contact them before you go to the field, how many tubes they expect. And also check if they do have DNA and RNA extractions, do they do high-Cs or not? And also you should allow some time for the project onboarding, because we need to make MTAs with you to send you sample submission forms and so on. This I will also leave there. So why do we need so many tubes? Because, well, we're working with something that is non-model. So we try the DNA extractions, what we think might work. If it doesn't work, we need to test another one. Then we need to test another one and say, okay, this starts working, but we need to modify to get even better material, and so on and so forth. So there is lots of troubleshooting and modifications when we work with non-model samples. Many of them we have never seen before, not just us, but scientists anywhere in the planet, right? You know, you're working with them. On onboarding, this is just an example of SciLife Lab. Very often we get email from user, hi, I want to send samples to you, pickup arranged for Tuesday, and I need your address. I said, that doesn't work. So we need to have this information in our portal so we can parse it to the LIMS. We need to make the MTA agreement. So your sample should get a code, because we work with hundreds, in some weeks, thousands of samples in parallel. We need to make sure that we can reliably identify your samples all the way through the process. And for that, we need to have the electronic tracking system in place. So that usually takes a couple of days. So to take home everything, talk to your providers, sequencing centers, they are your best friends. long-read technologies, they're very sensitive to DNA quality. So if your organisms consist of A, T's, G's, and C's, it doesn't mean it will sequence. So, and then nothing is standard when it comes to sequencing non-model organisms. Very often the sequencing personnel that works with your samples is overlooked. So I gave you a little bit of the flavor of the troubleshooting and R&D that are being done by the sequencing facility professionals. But it's very sad to see that very often we are either not acknowledged or not even offered publication or co-authorship, because there is lots of work going into making the genomes work. The genome is not a target, it's a tool, and we are here to help you to produce the tool to enhance your studies. So this will be the thank you, and my very last slide. We have the long-read meeting in Uppsala. Abstracts are open October 23, and we have 3 different breakout workshops that can be of interest to the ERGA community. First of all, in terms of bioinformatics, we have best practices in reference genome assembly, led by Henrik Lantz and Kirsten Howell from Sanger. In terms of science, Jakob Höglund is organizing biodiversity breakout sessions separately. And on the lab, Alve and myself, we are organizing workshop on best practices in genome sequencing. It's not only about the optimization of the sequencing, but also optimization of DNA extractions and sampling and things like that. So warm welcome, everybody, and I will stop talking.